In this lecture, I'm going to talk about possible mechanisms for parasite neutralization of Plasmodium falciparum asexual blood stages. And as can be seen here in this slide, that there are in this one actually the several mechanisms that mediate killing of malaria parasites, and these mechanisms could be antibody mediated or could be cell mediated or could be due to cooperation between antibodies and cells to mediate killing of malaria parasite. And actually the first mechanism, it is antibody-mediated phagocytosis of whole infected erythrocytes or merozoites, as can be seen here. These merozoite and these the whole infected. So these specific antibodies are raised against some molecules on the surface of the red blood cells. So these molecules are derived from the malaria parasite, and as you know that when the red blood cell get infected, some molecules get expressed on the surface of the infected red blood cell, but these molecules are related to malaria parasite and not to red blood cells. For example, Plasmodium falciparum membrane 1 BFEMB1, Plasmodium falciparum erythrocytic membrane protein 1. So these antibodies may be due to they are raised against these proteins. So when they bind to specific antigens, they will opsonize the infected red blood cell to the monocytes and mediate phagocytosis of the whole infected erythrocyte or merozoite. And also phagocytosis, it could be antibody independent through some adhesion molecules, as can be seen here. This infected red blood cell gets phagocytosed by monocyte without antibodies. So the possible also mechanism for parasite neutralization of Plasmodium falciparum asexual blood stages could be through complement-mediated phagocytosis. And as you know that monocytes express some complement receptors. For example, complement receptor number 1, which are specific for C3b or C4b. So binding of infected red blood cell to this complement receptor, this mediate also killing by means of the complement. So also one of the possible mechanism is through the tumor necrosis factor alpha and nitric oxide. These chemical intermediates actually they come from activated macrophage. This macrophage derived from the monocytes, and so tumor necrosis factor alpha and nitric oxide, they have parasiticidal effect on the intraerythrocytic parasite or on merozoites, and as can be seen here. So in these mechanisms, antibodies interfere with the binding of merozoite to the red blood cell. So here antibody mediate inhibition of merozoite invasion. And as you know that antibodies they work by 3 ways: opsonization, neutralization, and complement activation. And also here the antibodies, as can be seen here, inhibit intraerythrocytic parasite development by binding to some antigens on the surface of the infected red blood cells. And similarly, the antibodies, they have the capacity actually to enter through the parasitophorous duct. And as you know that merozoites do not penetrate red blood cell, but they press and invaginate the red blood cell and create a bed. It is known as parasitophorous bed. So antibodies through the duct of the parasitophorous bed can enter and interfere with the development of the parasite within this parasitophorous bed. And here in this mechanism, actually antibodies, they find their way to intraerythrocytic parasite just before rupture of the schizont. So this also interfere with development of these parasites. Thank you very much, Professor Ahmed Bolad.